Namaste friends, welcome to Bano Biology Center for True Learning. Here I request you to subscribe our YouTube channel Bano Biology Center for more videos and press the bell icon for latest updates and notifications. In this video, we will learn the concept fertilization and implantation. What is fertilization? The process of fusion of a sperm with an ovum is called fertilization, or the process of fusion of a male gamete with a female gamete in human females is called fertilization. Before fertilization, the insemination should be occur. What is insemination? The releasing of sperm by a male partner into the vagina of female partner through his penis, called insemination. The insemination happens here. The sperms are released in the vaginal part. The motile sperms swim to reach papillary-isthmic junction of fallopian tube through cervix and uterus. This is the place where sperms are deposited by the penis. From here, the sperms move through the cervix. This is the cervical region. Through the cervix, the sperm enters the uterus part. This is the uterus part. From here, the sperms enters the isthmus ampullary region. This is the junction of ampulla and isthmus part. This junction is called ampullary isthmus junction. The ovum released by the ovary also reaches this region. First, suppose the ovum is released from this ovary, immediately it is catched by the fimbriae. From fimbriae, the egg also moves towards the uterus and reaches the isthmus region here. I mean, both the sperms and ovum reaches the ampullary-isthmic region for fusion purpose. Here, ampullary-isthmic region is the actual place for fusion of both the gametes. I mean, for the fertilization. So, what is the fertilization place in the human female reproductive system? The ampullary-isthmic junction is the fertilization site or place. Here, fertilization only occurs when sperm reaches this junction at a time. Here, if fertilization need to be done, both the gametes, male gamete sperm and female gamete ovum reaches this junction simultaneously. I mean, at the same time. Otherwise, the fertilization cannot happen. All copulations do not lead to fertilization and pregnancy because of the reason. If both the ovums are unable to reach the ampullary isthmic region at a time, the scope for fertilization is almost nil. Then capacitation takes place in female reproductive tract. It is a very important word. Capacitation. What is capacitation? The removal of the mucus substance formed on the acrosome part of the sperm by the secretions of female genital tract is called capacitation. Actually, acrosome part, this is the acrosome part of the sperm has some receptors. When the secretions of reproductive tract of the female clean the acrosome, these receptors become exposed and sperm will become activated. Simply we can say that capacitation is nothing but the activation of sperms is called capacitation. It takes place in the female reproductive tract. A sperm comes and has contact with the zona pellucida layer of the ovum. Firstly, the sperm reaches the zona pellucida layer. This is the corona radiata layer. Inner to this zona pellucida layer. So after crossing the corona radiata, the sperms reaches the zona pellucida. Then immediately the acrosome pod releases the sperm lysins. This is called a acrosomal reaction. When sperm lysins are released from the acrosome, this phenomenon is called acrosomal reaction. So what type of sperm lysins released by the acrosome? Here are these. One is hyaluronidase, another one is corona hydrolytic enzymes, the third one is zona, lysin or acrosin. These three are the important sperm lysins released from the acrosome part of the sperm. Let us see the functioning of each sperm lysin. The first one, hyaluronidase. It mainly removes the hard substance of corona cells. There is a cementing substance between these corona radiata cells. That cementing substance can be removed by the hyaluronidase enzyme. It loosens the radial cells. Then another sperm lysine is corona penetrating enzymes. It mainly digests the separated corona cells. After separation of the corona cells by the hyaluronidase, the corona penetrating enzyme digests all the cells, make the disappearance of the radiatory cells completely. Then finally this layer will disappear. After digestion of the corona radiata layer, the sperm reaches the zona pellucida. Here, acrosin lysin, sperm lysin helps in digestion of the zona pellucida layer. This is the zona pellucida layer. It is digested by the acrosin, which is the sperm lysin. Then the plasma membrane, which is also called vitelline membrane, experience the depolarization. So when sperm reaches the plasma membrane, this is the plasma membrane. When the sperm reaches this plasma membrane, when it touches it, the depolarization occurs. The depolarization is must for preventing the polyspermy. What is polyspermy? The prevention of all the sperms enter the egg is called polyspermy. Only one sperm is going to successfully enters the egg and fuse with the nucleus of the egg. All other sperms are blocked from the entry into the egg. This phenomenon is called polyspermy. The polyspermy is usually stopped by cortical granules. These are the granules present in the cortical region called cortical regions. The blocking of polyspermy is mainly due to the functioning of these cortical granules. Then meiosis II finishes in the oocyte as the sperm enters the plasma membrane. When the sperm cross the plasma membrane, the stopped meiosis II at metaphase again restarted and will finishes soon. Then two unequal cells are formed. I mean meiosis II is also results in the formation of two unequal cells. One is a second polar body, another one is haploid ovum, which is called oocyte. The haploid cell and ovum formed due to the meiosis II is called oocyte. Then egg lacks centrioles of its own. The egg doesn't have the centrioles. Here the centrioles are required for formation of the spindle fibers during the first mitotic division. Here the egg lacks the centrioles. This picture is very clearly showing us about the entry of the sperm into the egg after crossing the layers one by one. Here the first, the sperm touches the corona radiata. The corona radiata layer will disintegrate or digested. Then sperm reaches the middle layer zona pellucida. It is also discarded by the acrosin. After that, it enters the pre-vitelline space and also to the vitelline membrane, that is plasma membrane. Here the plasma membrane also will be discarded. Then the sperm enters the egg. Sperm makes contact with egg. Acrosome reacts with zona pellucida. Acrosome reacts with pre-vitelline space. The plasma membranes of sperm and egg fetus. Both the plasma membranes of egg and the sperm fuses. Then sperm nucleus enters the egg. This is the actual process of fusion of gametes. Next, the sperm contains two centrioles. We came to know while we were discussing the structure of sperm. One is the distal centriole, another one is the proximal centriole. Here we want to talk about distal centriole. The distal centriole of the sperm divides to form two centrioles. These two centrioles are required for formation of the spindle fibers, as the egg doesn't have the centrioles. Then haploid nucleus of the sperm fuse with the haploid nucleus of the ovum to form diploid cell called zygote. Here, this is the haploid nucleus of the egg. This is the haploid nucleus of the sperm. Both these fuse together to form the diploid cell, which is called a zygote. Zygote is the diploid cell, which is result from the fusion of both egg cell and sperm cell. Here, the mixing up of or blending up of the chromosomes of the spermatozoon and the chromosomes of the ovum is called karyogamy or amphymixis. The sex of the baby is decided at this stage, I mean at the zygote stage, whether developing fetus will become male or female will be decided at this. Thank you for watching this video.